welcome to Innovate and React. I'm super excited to have another podcast as my guest today, Alexandra from the PhD Experience podcast. She's a biochemist, and today we will be talking about how the power station of the body works and how they import proteins and function in general. So maybe let's start with your background. What brought you to biochemistry?
Yeah, gladly. Hi, everyone. I'm so glad to be here. I wanted to say this. What brought me to biochemistry? It's actually a really fun question. Since I was little, my parents actually wanted for me to become a dentist.
The classic.
The classic. Thankfully, they weren't the type of people to pressure me into this, but they just thought it's a nice kind of career to have where you combine work life balance, have a nice income, and so on. So, this is what I thought that I wanted to do for a long time. And then we moved to Germany when I was 16. And then this was a whole new world for me. Nevertheless, long story short, being new in Germany and having learned German, fresh when I came here, I finished my high school here. Nevertheless, my grades were not, 1.0 or 10 equivalent in other countries to get into dentistry studies. So, as a backup plan, because I was expecting this would happen, I actually applied for some other science studies, and biochemistry was one of them. And then it happened that I got in at the university in Ulm, which I'm really proud of.
Was there a cap at the time when you applied?
Yes. Yes, actually there was. So I was kind of lucky to get in.
Yeah, great.
I think science is just this kind of universal language in a way. You don't need to speak a specific like English, German, Spanish, whatever. You just understand science, and then you kind of understand each other and also how the world works. So for me, science is just this kind of really nice place where everything makes sense.
Definitely.
Until the PhD.
It's also great. It's not within the borders of countries. You can just,
Exactly.
scientists don't really care about that.
Exactly. And this is what I love about science and the fact that we base a lot of our work on collaborations, on networking, on international connections. Like there's so many papers, scientific papers out there of labs, from 3 different countries or more working together for one project, which I think is really beautiful. Of course, what happens backstage and so on is another story, but together to get all of this out like a paper and contribute to the field of science and bring progress into it, I think is just really great.
Nice. And then you continued doing your PhD somewhere else, right?
Basically, I did my bachelor's in Ulm, I did my master's in Ulm, also in biochemistry. And then after the master's, I had 2 different jobs, one in academia and one in industry, because I didn't want to do a PhD at the time for different reasons. And then after my second job, which was in the industry, I realized that I wanted more and I wanted to stick with science a bit longer, and I was missing this research aspect and the freedom that you get being in a lab at the university. So that's when I applied for a PhD position at the University of Tübingen, and this is where I am now. I am in my 3rd year and I am studying mitochondrial biogenesis and dynamics.
Biochemistry is a really interesting connection between biology and also sometimes medicine even and chemistry, and it connects a lot of dots, I would say.
Definitely, but in Ulm we used to have this joke that biochemistry is the black sheep of chemistry, but also black sheep of biology.
Yeah. Yeah.
So, yeah, doing everything and maybe nothing, I don't know. Yeah.
Yeah, in Dortmund we also had actually a lot of chemical biology. That's the same, I would say.
Yeah.
It's a bit more on the biology focused somehow, but also they are not biologists directly, but they're also not chemists. And
But I think that's nice because I kind of identify this with my personal life experience so far. Because I was born in one place, raised in another, and then moved to another, so I cannot identify with one nationality. And having different disciplines within science become one, like chemical biology or physical chemistry or whatever.
Mhm.
I think that's really great.
Yeah, definitely. So you said that your topic is mitochondrial biogenesis.
Yes, correctly.
So since biology is not like that familiar to me and the audience, maybe we can start like from a bit more back than usual, I would say. So the mitochondria, as I remember from biology class, they are the power plant of the cell and give energy. And yeah, how do they work and what is their responsibility in the cells more detailed?
That's such a great question. So yeah, everyone knows mitochondria are the powerhouse of the cell, definitely, but they are actually performing so many more functions that are not highlighted as much. Nevertheless, they're really substantial for just homeostasis within the cell, and we're talking now not only human cells but also other organisms. So any cell that contains a nucleus usually has mitochondria in it, right? And mitochondria are there for the energy, like you said, but then they perform other functions. The heart and the brain, they are tissues in our bodies that need more energy. So this is also where we get a bigger number of mitochondria, but not only for the energy, but also, for example, for the calcium release, right, when we need it in order to have muscular function.
Interesting, I didn't know that.
Exactly. And calcium is also used as a kind of signaling chemical. So in our neuronal cells, in our brain, you can have also release of calcium, and then different functions will start happening, and different receptors will react to this release of calcium. So this is another thing that mitochondria do. On top of that, they're also responsible a bit for the metabolism within the cells. So sometimes, maybe our cells are facing some conditions that are, let's say, harming to them or dangerous to them, and then someone needs to say that this cell or this group of cells actually needs to die, right? Like it's important for the cellular processes that we get rid of bad cells, so to say, right? So then mitochondria also helps in regulating this process by releasing specific proteins out there that signalize, that okay, now we have to start the so-called apoptosis pathway, which means basically starting to die. So the death of a specific cell or a group of cells in order to make place for either new ones, to regenerate or something else. Yeah, and on top of that, I actually brought a fun fact with me. So I don't know if many people know this, but mitochondria are also capable to produce heat. Of course, not in all tissues, but in, I think it's brown fat tissue, they are able to produce heat through a process that is called thermogenesis, which is also needed in this specific tissue. I don't know all the details because this is not my area, but I thought it's a fun fact to know.
Interesting. So they can also be heaters, not just the power, but also heat.
Exactly. And of course, in humans, I mean, I guess majority of biochemists know this. They help break down fatty acids. So, from the intake of the food, from nutrients and so on, someone needs to break down also, next to the sugars, also the fatty acids, so that our body can process them and use them wherever they're needed. And then mitochondria are also responsible for that. Yeah.
So they also convert energy, because as I understand that fats and sugars are energy sources.
Exactly.
And then the body will make glucose out of the fats. Is that what the mitochondria do, or is that somewhere else?
Not so. Mitochondria, not as far as I know. So they do take the glucose and they basically transform it into ATP, which is this kind of energy currency for the cell. So our bodies need this ATP to perform. But on top of that, it takes these fatty acids, which are usually long chains of carbon and other chemical elements, and basically break them down into smaller parts. And then these parts can be used either for the mitochondria itself or in other places within the cell.
Interesting. So they have a lot of different functions for the cell and are really crucial for the survival of these cells, or the death apparently also. And to keep them functioning, the cell needs to bring the building blocks for the mitochondria into the mitochondria. And this is what your research field targets, as I understood.
Exactly. So I will just give a brief introduction to bring everyone on the same page. So mitochondria is actually the only organelle within the cell that has its own DNA, right? Nevertheless, because they're small organelles, there's not this capacity to really produce all of the proteins that they need. This is also kind of the clue that scientists used to show that mitochondria actually joined the eukaryotic cell much later, and that their ancestors are bacterial cells. Then when the mitochondria came to the cell, basically the biggest part of their genome went into the nucleus and basically joined the genome of the cell. So majority of mitochondrial proteins need to come from the nucleus past the so-called cytoplasm, which is basically, if you think of the cell as a house, right? Each organelle is a room, and then the cytoplasm, this is the hallway, basically, so to say.
Yeah.
And then, to go from one room to the other, you need to pass the hallway. So the proteins from the nucleus need to pass the so-called cytoplasm and then go to the mitochondria. Now, this sounds quite easy. Nevertheless, you have to keep in mind that mitochondria need constantly new proteins getting delivered to them. On top of that, we have so many more other organelles within the cell that, are kind of living from proteins that come from the nucleus.
Yeah, that sounds very complicated actually. When you have, you produce a protein and then this protein has to go somewhere, and they are all produced in the same place, the nucleus.
Yes.
Right? Or no, are they produced in the nucleus?
They're not really produced in the nucleus. So the coding sequence for these proteins lies within the nucleus, and then this specific coding sequence needs to be written, so to say, in a different code, which is called transcription. So from the gene you get the mRNA, which maybe a lot of the listeners know from the COVID vaccines.
Yeah.
And all of this is still happening within the nucleus, right? So then this piece of mRNA, this new code, needs to be exported out of the nucleus, go to the hallway, so to say, and this is where it will get written again in a different code, which basically will give us the protein. Yeah.
And then the protein has to go somewhere where it's supposed to be.
Exactly.
And this sounds really interesting on like how can this be possible? This is also a thing where biology and chemistry is very different, that such super complex processes are really difficult to understand, I think. Or it seems much more complicated, and you cannot just rely on a basic concept. In chemistry, you have a lot of concepts that you learn in your studies, and then you can apply them, and really like what works for one reaction should also work for the other reaction.
Yeah.
And in biology, that always amazes me, that how there's this one way this works, and it's really a miracle that it works. And yeah, evolution over time brought it up.
Yeah, I think it depends. I mean, if I think back in my uni time, because we did a lot of chemistry, we did anorganic, physical, organic, and so much more. I remember feeling overwhelmed by all these concepts. I was good in redox equations, like I really like those. But when you asked me, oh, which process are you going to use to produce ammonia? I would tell you I have no idea. Maybe the first one that comes into mind, and don't ask me why. So I think it's a bit mutual, but I think it also has to do with, of course, tendencies and what you like and inclination. And I think for biology, we're taught these concepts, and this is kind of like in history class, you're taught how kind of certain things work, and then you base everything else, the rest of the knowledge that you acquire a long time, on top of that. I don't know if that makes sense, but this is how I see it for me. Yeah.
That's why also the first year in the bachelor is very, very difficult. And then the further you get, the more it makes sense, because all the puzzle pieces at some point add together, and then you can really understand it better.
Exactly.
So yeah, that okay, then I understand that since you have the biology or more biology background, that this probably makes more sense to you. But when we had also the bioorganic chemistry classes, that was just for me very like miracles. Okay, now let's come back to the mitochondria. We have discussed that the nucleus gives basically the recipes for the proteins that are synthesized in the hallway of the cell. What was the name again?
Cytoplasm.
The cytoplasm. And then from there, they have to somehow find the right room, the mitochondria, and get into the room.
Exactly.
Which I think is also for the mitochondria, I don't know if all the other organelles also have that they have this double membrane.
So mitochondria is actually, I believe, I don't want to be absolute here, but it's, if not the only one, definitely one of the few ones within the cells, because there are many different organelles, and there are some whose shape changes according to different events that happen in the cell. But to my knowledge, mitochondria is either the only one or one of the very few that have this double membrane, like you correctly said. They have this double membrane, which is very important for compartmentalization. And this basically helps them run different processes at the same time without disturbing each other. because you have different redox reactions happening, you have different pH levels, in the different compartments.
Within the mitochondria, there are different pH levels in these compartments, or in the cell?
In the cell in
Yeah,
general,
okay.
but for example, between the two membranes within mitochondria, you have this process where you have these amino acids called cysteines, and they are known to build disulfide bonds, right? And when you build this disulfide bond, this is when you perform, like, this is when the sulfide is being oxidized, right? So basically, you have this media where redox reactions are happening, but you still want to keep these media conditions away from another compartment within the mitochondria where you don't want, I don't know, free electrons running around, so to say. Yeah.
That makes it even more complicated. So they have to find the right room to stay in this analog. Then in the room, they also have to go to the right corner, I would say.
Exactly. And because this is not complicated enough yet, when proteins are being written in their final code, so to say, right? So when the protein emerges from the hallway, you can picture it as a straight string, but then this is their, so to say, their primary structure, right? But proteins are known for reaching tertiary structure. So this means that they need to get into a specific shape and fold into that specific shape. At the same time, we have some other machineries within the cells in the hallway that make sure that proteins stay in this kind of primary structure, in this string form, until they reach their end room and compartment. And only then can they take their final shape, which will actually confer them their function.
Oh, so the function depends not only on the proteins, like the amino acids they are made out of, it also depends on the structure.
Exactly.
And it must not be in that structure before it enters the mitochondria, because then it cannot pass
Exactly.
the membrane.
Exactly.
Okay, yeah, that's a really complicated process. So how do these proteins then get into the mitochondria through the membranes?
That's a really good question. And I mean, a lot of knowledge is already, elucidated about the final steps of, proteins passing the mitochondrial membrane. So, on the outer part of the mitochondrial membrane, we have different protein complexes sitting there. So, protein complex is just multiple proteins, that kind of together they build this complex, and then together they perform a specific function. So then on the outer membrane of mitochondria, we have these complexes that kind of make sure to recognize specific signals that are attached to the proteins, and if the signal matches what these complexes are kind of expecting, so to say, to recognize, then they get to pass through. And then because we have all these different subcompartments within mitochondria, then they still need to pass more recognition mechanisms, so to say, until they reach their end destination. So it's not enough to pass the first gate, in the outer membrane. In the second membrane, so the inner membrane, you have to pass then a second gate, and then depending on if you want to go as a protein within the inner membrane itself, or if you want to go into the very deep of the mitochondrion, which is called the matrix, this depends also on different machineries to recognize and make sure that these proteins reach where they're supposed to go.
So they have kind of address labels on them.
Exactly.
That getting recognized by these machines, molecular machines.
Exactly.
Right? Yeah. So this is also all kind of then in this very deep molecular level, also a lot of chemistry. I guess that's why it's biochemistry in the end.
Exactly.
Right? Yeah, interesting. I have actually never really thought about it this like in this depth to reach this molecular level. So interesting. So there are these markers which are recognized. How do they get recognized?
So the thing is, it depends on where the protein is meant to go at the end of the day, right? So the deeper you go inside the mitochondrion, meaning if you are supposed to go to the matrix, or if you're supposed to go to the inner membrane, or maybe even in some cases between the two membranes, the proteins have at their beginning a so-called MTS, which stands for matrix targeting signal, which basically consists of positively charged amino acids. So then the gates from the mitochondrial membranes are able to recognize this because the protein enters the gate with this signal first, right? Makes sense.
Okay. So they basically grab on everything that has this marker on it.
Exactly. And then when the protein reaches the matrix, at the very end stage, there is a specific enzyme or protein, however you want to name it, that makes sure to cleave off the signal. So the signal is cut out from this protein, and then this protein can take its final shape, this tertiary structure, and then it can perform the functions that are required from it.
Interesting. So different ways of getting into the cell, and then once they reach their destination on their own, they form this tertiary structure once this messenger tag is cleaved off by this enzyme. And then they are ready to do whatever it's supposed to do in the mitochondria.
Yeah.
Interesting. So these then do all kinds of functions. And what is the problem now your research is targeting?
My research is actually based on a pathway which is called ER-SURF. It's a really fun name and a very fun project too. So ER-SURF stands for ER surface mediated transport. Now, to just make it a bit more simple, break it down a bit, the ER, endoplasmic reticulum, is just another organelle or just another room within the cell, right? So every organelle that we have within our cells is important for various reasons, and we have it there because evolutionary, right, it made sense to keep it there, perform its function, which led ultimately to our survival. Now, for a long time it was thought that this compartment, the ER, it's competing with mitochondria in terms of targeting of proteins, right? Scientists thought for a long time that these two are not collaborating with each other, but they're rather competing for who's going to get the proteins. Now,
Oh, okay.
exactly.
So also was at that stage when they thought they were competing not known that there were these labels, for example, which tell the cell on where to bring the proteins?
It was known. However, the thing is, The targeting signal for the ER has some similar characteristics as the one for mitochondria. The one for the ER is a bit longer. Nevertheless, in the past, there have been multiple cases where scientists had observed proteins that belong to the one compartment being localized in the other one. So this is why they thought these two are competing with each other for who's going to get the protein. And back in 2018, so wow, it's already 7 years ago, a scientific group from Germany, actually, from Kaiserslautern, they discovered that the ER and mitochondria are actually cooperating with each other and they're working together. And they found this under this ER-SERV pathway, which they named themselves like this. So basically what they discovered in their paper was that proteins that normally belong to mitochondria, and by different reasons they mistakenly go to this ER compartment, right? Instead of them getting degraded and getting thrown away, so to say, they actually saw that these proteins got a second chance to be brought back to mitochondria.
Oh, nice. So it's like a neighbor bringing your package back.
Exactly. That's such a nice metaphor. Yeah, exactly. So this is actually my project, trying to understand this pathway better, trying to make also the difference between proteins that come back to mitochondria, but also proteins that actually get thrown out and don't get the second chance. Trying to understand why is that so, which factors are involved there, what is happening, and on what is all this dependent.
And how do you look into this? Can you really isolate those proteins with the markers on them and then see, okay, what changed now on the marker, or at what parts of the protein are you looking at to see what went wrong?
So the way I'm doing it is actually, so because this is fundamental research, right? And working at a university, we're not really equipped with the most, state-of-the-art machines and technologies. So we do a lot of the things old school. So basically we grow a lot of yeast cultures because in my lab we work with yeast, because yeast is classified as the same type of cell as human cells are. Of course, there are differences between us, definitely, but there have been many instances in the past, where someone discovered a protein or a pathway that happened in yeast, and then by doing some computational studies and so on, finding similarities between proteins and different organisms, people could show that this process that takes place in yeast takes place in humans as well. Of course, in humans, much more complex, with many more players involved into this, definitely. But all in all, yeast is a good organism. It grows fast, it's cheap, genetic mutations, modifications are very easy to perform. And in humans, you have the problem that you have two sets of chromosomes, right? The ones from your mother and the ones from your father. So then if you want to, let's say, delete one gene, you would have to do it in both chromosomes, right? But in yeast, you have the option to have yeast cells with only one set of chromosomes, which means you perform the deletion process of the gene only once.
Yeah.
And it's much faster.
That makes it much easier. Yeah.
Exactly.
And you can probably also then do less changes at a time, which makes it easier to trace back what comes from where.
Exactly. It's way more stinkier than human cell culture. But this is something you get used to. And if you like, bread or drinking beer,
Yeah.
working with your yeast is just for you. Yeah. So basically, we grow yeast cultures, and then we use big centrifuges that basically reach very high speeds, right? And then we take advantage of this fact and spin down different fractions of our yeast culture. And we treat the yeast culture, with some enzymatic cocktail to break down the cell wall. And then we also use some mechanical stress, some mechanic power. We douse the culture in a douncer to help, the cell wall breaking open. And then we take the different fractions, we centrifugate them at different speeds, and this is how we isolate the different compartments from within the cell. So then you can have, for example, isolated mitochondria, you can have isolated nuclei, you can have isolated ER, like I mentioned previously.
Oh, interesting.
Exactly.
You can really isolate them. I thought that you would just have one soup and you cannot really isolate them because they're like all so complex and somewhat large molecules that really have not that much of a difference from like a bigger perspective.
Yeah.
I know when I am thinking about my research and I try to isolate the different side products from my reactions, that always was really a pain because they were sometimes very similar and then it's really difficult to get them isolated. And I thought in the cell, since you have so many different molecules, this would be a mess.
Actually, we take advantage of the different rooms within the cells, right? The different organelles. They have different densities because some of them are bigger, some are smaller, some are packed with a lot of proteins. Others are, maybe lacking proteins. Others get formed within the cell when stress conditions appear. So you have all kind of these different factors, but what we are basing our experiments on is density mostly, so we do these differential centrifugations at different velocities, and then okay, if you centrifuge at 200,000 G, for example, you will get the ER fraction. But if you centrifuge at 20,000 G, right, so 10 times less, you get mitochondria.
That's very interesting. Because on a chemist point of view, gravity rarely plays a role because the molecules are too small, so it's not playing a role because of the solvents you're working with or whatever. The interactions are just bigger and then that doesn't play a role. But this then can be compared almost to like a distillation where you just heat up and then get different fractions
Exactly.
from different temperatures, and there you can differentiate the organelles based on their mass and density.
Exactly. Cool. And then you take these organelles, and then you can perform so many different experiments with them. But usually the first step is to measure the concentration of proteins you have within this fraction so that you know the amount you're working with, right? So you want to also normalize your working procedure. You want to have all the samples, all the fractions that you're isolating to the same standard, right? So imagine it as a marathon. You want everyone to be on the same line, right? So that you can compare at the end of the day what is what. So then we measure the concentration, and then you can do many different applications in experiments with them. But one very basic one that our lab does daily, for sure, is the so-called Western blot, which Long story short, basically means you take a protein sample like this one, you run it on a specific acrylamide gel, then you get a separation of all these proteins within your sample based on their mass, and the separation happens with the help of electric current. And then with the help of, antibodies, which bind to these proteins, you can then identify specific proteins that are present or absent in that fraction that you're looking at.
And that's how you know which proteins are in which
Compartment.
sample, and based on that you can find out if these proteins have these messengers on them, I guess.
The thing is, we wouldn't really be expecting to identify that kind of targeting sequence from the beginning, because this is a constant dynamic process that happens, and actually quite fast as well. So I think the probability to catch that is quite low. Nevertheless, there is a field within biology, biochemistry, what you want to call it, where scientists are really occupied with taking singular proteins, and kind of reconstructing them in a different organism, and then purifying them, and then really, really looking very detailed and closely into the sequence of it. And then, they can say, oh, we have more positive amino acids here because of that, or the protein is not folding correctly because of another reason. So, yeah. Unfortunately, we don't do this in my lab, but yeah.
So how do you find out then, after all these separation methods, on what went wrong, why it didn't go directly to the mitochondria?
So the thing is, as every scientific project, right, you start with a hypothesis that usually is based on a lot of research from the past. My project concretely doesn't have much research done on it. There are, I think, maybe 4 to 5 papers in total out there about it specifically, and the rest is just hunches and kind of poking in the dark, hoping that this is, the mechanism you've been looking for. So a lot of the things, I would say maybe 85% of the things I've done so far for my PhD have been kind of educated guesses, hypotheses based on previous research, trying to connect the dots in a logical way, and then kind of going for it. So then you would create, for example, different yeast strains, with mutations. So, for example, if I believe that protein A is responsible for bringing, let's say, in general, other proteins to the mitochondria, I can then go and delete protein A from yeast, and then perform a centrifugation, separate the different fractions, then put this on that gel I mentioned previously, put the antibodies on the on this gel afterwards on the blot, and then check out the difference in comparison to the healthy yeast strain, so to say. In biology, usually this is called the wild type, and you can use it actually for everything. So you always compare to the wild type, to the natural one, so to say.
Ah, okay.
Yeah.
So you try to first see what proteins there are and what you guess what function they have. Then you delete them from one culture and see if, for example, when you think they would bring the proteins to the mitochondria, and when in your second run there are no of these proteins in the mitochondria, you can assume that this protein is responsible for bringing it from wherever it's produced to the mitochondria.
Exactly.
Oh, that's very cool. And I think also this really trial and error kind of research where you have to make guesses and then probably also brainstorm a lot with your colleagues and then do these kinds of experiments, and then in the end, when something actually works as your hypothesis said, then yeah, I guess that's a great way.
That is a great way when it works. Like you guessed, I have had really a lot of hypotheses so far. And that's like a thing I really love about science. But when you kind of stay stuck in a place, it's also what makes me hate science at times. That a lot of the times it's just guess after guess after guess, and your constant kind of story that you try to build in your mind about your project keeps changing because the results keep showing you this is not working, this is not it, this is not, so you have to keep going. And of course, you have to do literature research and get new ideas from either other researchers, or you can also look if the process you're kind of researching on, if it exists in maybe a different organism, what is known about it there, try to see some parallels, and then maybe you can use something from the other organism for yours. So it's a lot of kind of connecting dots. Maybe sometimes even it goes in the direction where they're not really dots to connect, but you make them be there. And then, of course, sometimes this can lead, to a successful paper at the end, but a lot of the times it can also just lead you in the complete opposite direction. And then you kind of, lose some time, right? Because time is really one big aspect of the PhD. But at the end of the day, it's all an experience, and I don't think that you can, I don't think that you can blame someone for taking the wrong guesses, right? Because let's say if your supervisor knew the answer, at least they would guide you there, right? They wouldn't let you go in the complete opposite direction. So, yeah.
But that's research that you basically have a lot of results that are just negative results is maybe the wrong word, but it's like not in favor of your hypothesis, but it still gives you more understanding of the whole concepts, because you can say, okay, this didn't work. Maybe you can then work on your hypothesis and get more insights also because of these results that didn't go the way you wanted them.
Exactly. Yeah. Exactly.
So when you have this fundamental research, is also in the previous episodes, there were some fundamental research where we discussed that it's not always direct application possible, but it will lead to a lot of new insights and new research building on that, and will be the basis for some applications in the future. Where would you see future applications of this kind of research you're doing? So what problem could that help understand better?
Yeah. So the thing is, this is still quite fundamental, definitely. Mm-hmm. Nevertheless, there are quite some diseases, some genetic deficiencies out there that kind of, they exist out there because of problems with targeting, of proteins to the right place. Mm-hmm. So basically, if one could understand this process a bit better and then realize the factors that influence this and kind of lead this process and make it happen, you could have maybe individuals, humans, who have specific mutations in their genes, that maybe make this specific key player for the pathway be knocked out, be not functioning. And then with, let's say, targeted gene therapy, you could take this specific gene, try to implement it into this specific individual's genome, and then try to make their bodies start producing this protein, and then hopefully make the individual healthy again. Nevertheless, I have to say that usually people who are born with deficiencies in the mitochondria don't really have a long lifespan, because mitochondria is so important, to produce the energy. It's what kind of keeps us alive, right? The food we take in and the oxygen we breathe, these are converted into energy in mitochondria. So if your mitochondria are defective and have a problem, it only makes sense that when you come into the world with such a deficiency, you won't have much time to live. So this would be one thing. So diseases that are based on this, transport process. And then of course you have cancer that can also happen from such processes.
Yeah, when the mitochondria don't function properly, and as you said in the beginning, tell these mutated cells to die, then they would reproduce uncontrollably.
Exactly.
I guess.
Or for example, Parkinson's. So mitochondria are involved there as well. And there are different reasons for Parkinson's, but for sure, like the targeted protein delivery is also an important one right there.
Oh, interesting. Yeah. So a lot of actually big health problems that humanity has.
Yeah.
Maybe it can contribute to that in the future. That is a very promising topic then.
I would love for that to happen, and honestly it's not even about my ego as a scientist, but just to see a project I've been working on kind of evolve into a different shape and gain importance. I think that would make me very happy. I mean, even if I don't end up with some big publication from my PhD, I don't mind this so much. But this time you spend with your project, and I think even after I finish my PhD, I will always keep going on PubMed and checking out ERSurf, what's new.
Yeah. Yeah, that's when you're passionate about the science, then it really also drives you to do all this work. And also what comes then after your research is then also communicating and talking about your research, your experience. And that is also where you started a podcast. Maybe we can introduce what that is about.
Yeah, gladly. So I have a podcast. It's called PhD The Experience. And I basically started it almost 2 years ago because I felt a bit isolated and I needed to connect with more people, to network, and kind of understand for myself how different PhDs, but also, post-PhD graduates are feeling about this whole process. How is their experience? Am I kind of going through something unique, or are people actually experiencing the same things as me? So this is how the idea for the podcast came. It was very random. I've told this story actually a couple of times already, on social media platforms, but basically the day I got it, I was in the lab. It was late in the evening, and then I went home, and this idea kind of lit up my motivational area in the brain. So I stayed up until late, and I was thinking, what music am I gonna play, and how the art is going to be, and how am I gonna structure the episodes, and who am I gonna invite. So yeah.
Yeah, that's great. A bit similar to how I started also, with this idea, and then you just can't stop thinking about it, and you just have to push it, and then after all, meet people, talk about their research or about their experience with their PhDs, and then share this with others.
Exactly.
Yeah. So yeah, with this, I would say we have end to this very nice discussion. It was really interesting to look into more biology and how chemistry is also a thing in biology. So thank you very much for sharing your research. It was really a pleasure to have you.
Thank you so much for having me. It was my pleasure being here, and I hope the audience didn't get too bored with all this biology stuff.
Yeah. It's always good to get a wider perspective and sort of think outside of the box to get new ideas and then maybe apply them to whatever you're working on. So it's always good to connect different fields.
Definitely.
Thank you very much for listening. Please hit the follow button on wherever you're listening. This really helps the show. Also, if you want to hear more about the PhD experience in general, then check out Alexandra's podcast, PhD The Experience. You can find the links in the show notes. If you would like to share your research in this podcast, then send me an email to podcast@innovateandreact.com or connect with me on LinkedIn. Until next time, keep innovating and stay curious.